Good afternoon and welcome everybody. I can just see that people are slowly filtering in, so I might just give it sort of another 15 seconds, half a minute, and then we'll get started. Fantastic. So I will open this webinar by first saying good afternoon and welcome to the AgriFutures Chicken Meat Program webinar series. I'd like to start with an acknowledgement to country. So AgriFutures Australia acknowledges the First Nations people of Australia as the traditional custodians of the lands and waters on which we live, learn and work. We pay our respects to past, present and future elders of these nations and any First Nations people on this webinar with us today. In particular, I'd like to acknowledge the Ngunnawal people of Australia, the traditional custodians of the lands and waters where I am located and hosting this webinar from today, which is Canberra. So to those of you who are new here, it's lovely to have you here. And to those returning to the series, it's also lovely to have you here and welcome back. So before we get into the nutritional nuggets of today's webinar, a little housekeeping first. We will be recording and are recording today's session, and we'll be making that available through online channels. As I do run through these housekeeping items, my tech support team will have an introductory poll that will pop up onto your screen, and that's with has some questions to gauge who's in the virtual room with us today, and so we'd be grateful for your responses. As this is a webinar, your video and audio functionality are disabled so that we can best utilize the Zoom real estate, or the screen real estate that is, to view the shared content of our presenters today. It also allows us to minimize background noise and distractions to our guests. So we will have time for question and answers at the conclusion of today's webinar content. But in the meantime, please use the dedicated Q&A chat function to ask any questions over the course of the webinar. We will be monitoring that, and I'll be asking our presenters those questions at the conclusion of their presentations. So a quick introduction to myself. I am Sarika Pandya, the Research Manager, part of the AgriFutures Levied and Emerging Industries team, and I specifically look after the Chicken Meat Research, Development and Extension Program. This webinar is the 5th in our series, and we like to host expert presenters to provide you with the latest information on topics that are both relevant to industry and the industry's research priorities. And today's topic is about broiler nutrition, or certain aspects of broiler nutrition. So the viability of the chicken meat industry relies on both the optimal health and welfare of animals, and that's supported by cost-efficient and environmentally responsible production systems. And feed accounts for more than 60% of the total cost of chicken meat production, which has made it and still makes it a major focus of research investment by the program. And this is captured as a key research priority within the program, which is centered around efficient and secure production systems. Our program invests in research and in projects via industry levies within this area, and we're really looking to drive evidence-based innovation in the application of nutrition principles. So I really hope you enjoy today's content. So I will begin by introducing our first speaker. So, and that is Dr. Reza Barekatain. Reza, you can start sharing your screen now with your presentation. So Reza is the leader of the Pig and Poultry Science Program at the South Australian Research and Development Institute, also known as SARDI. He's also an affiliate senior lecturer with the School of Animal and Veterinary Science at the University of Adelaide. He obtained his PhD in poultry nutrition, and his main research interest has been at the interface of nutrition and gut health, and that's with the aim to improve productivity, stress resilience, and sustainability in poultry production. And Reza is really passionate about researching nutritional strategies for both pig and, but more specifically to this webinar, poultry production, and the nutritional strategies for improving intestinal barrier function and gastrointestinal ecosystems. And that's as a means to alleviate detrimental implications of enteric disorders and stress-related factors that monogastrics will face during their production cycle. So Reza will be informing us today on protein source and selected amino acids for gut integrity of chickens. And with that, I'll hand over to Reza.
Thank you, Sarika. Good afternoon, everyone. First I'll thank AgriFutures for their invitation and support of this project, which I'm giving an overview about. This is a project that was supported by AgriFuture, and it was completed and reported earlier this year. So this is basically a quick overview of what we did in that project. Some part of it has already been presented as part of other webinars and conferences, but the idea of this presentation is to just give an overview. So in this project, we focused on reduced protein diets, basically looking at source of protein and selected amino acid for gut integrity of chickens. In chickens, similar to other species, when we look at the intestinal integrity and intestine in general, the core part of intestinal barrier function is epithelial cells that are linked together by a series of protein that we call tight junction proteins. The function of these enterocytes is that they facilitate selective absorption of nutrients, but at the same time, they control and prevent the entry of unwanted materials, bacteria, toxins, and so on. So we don't have follow-on issues from these unwanted materials, or basically we don't want to have bacteria translocation. So under normal conditions, this process works quite fine, and the absorption happening, and bacteria translocation, as I said, is prevented. But when we have factors like stress or disease or other factors that can actually compromise intestinal barrier function and increase the permeability, the integrity of these tight junctions are impacted, and as a result, toxins and bacteria and those problematic, I guess, stuff that exists in the gut, they can find their way into the body, and then obviously at the end, some of them will find their way to the blood and to the internal organs and cause the inflammation. So, and that will obviously reduce the performance and productivity of the animal, in this case, broilers. So this is the main part of this project. So we focused on the intestinal integrity, but in the context of nutrition and reduced protein diet. As a general introduction, we are interested in reducing the dietary protein because it has got benefits for environment, for welfare, and sustainability of production. But there are perceptions that if you reduce the dietary protein, it can improve gut health, basically based on the fact that it can reduce the undigested protein in the hindgut, and as a result, the proliferation of harmful bacteria or those that we don't see them as beneficial is less promoted. So in that case, it may have some gut health benefit. But the key question here that we looked at in this project is that how this perception is relevant in the context of poultry nutrition, in the context of reduced protein diet, because we don't simply reduce the protein, we supplement the synthetic amino acids, and we have other changes happening in the diet that may have impact on birds' performance and gut health. So in general, the objective was to see what is the impact of protein reduction when we supplement the synthetic amino acids compared to control and standard diets. And we also were interested to see whether there is any impact between difference between vegetable-based diet and excuse me, and one that has got animal protein, because in Australia we still use some level of animal protein in mainly meat and bone meal in some cases. And also the other part of the project were related to investigation of the effects of some selective amino acids that were perceived to be important in this context, glycine, serine, and threonine. And also branch chain amino acids for their impact on intestinal integrity. And we were interested to see if these amino acids and these diets can actually counteract or interact with gut barrier dysfunction model that we used in this project. So the very first experiment, we looked at reduced protein diets, 2 levels of reduction. As you can see in these slides, the percentage was fairly extreme in compare with what is used commercially. We just wanted to see whether we can actually impose the impact on the birds to see a response, and then fine-tune from that. So we had a trial that lasted 42 days, and we had 2 parts in the trial. We had grower and finisher phase of feeding, and from the day 14 to 21, we had a challenge study using dexamethasone as an agent to cause leaky gut in the birds by injection of birds 3 times on day 14, 16, and 20. And we had a permeability test on day 21, which was a standard FIT-C test that we did by oral gouging the birds based on their body weight, proportionate to their body weight, and then followed by time-dependent blood collections. And we also collected samples for gene expression and microbiota analysis as well. So when we look at the response for the intestinal permeability, which is a key measure for the intestinal barrier function, one of the key measures, we saw an independent response to reduced protein diets. And the diet that had the lowest level of protein had the highest intestinal permeability. And that was independent of the DEXA challenge that we used in that study. So we have published that results of that study. Last year, we published it in Animal Nutrition, and yeah, that's available online in an open access for anyone who may be interested to look at those results. But here I highlight some other results from that study, and then move on to the following experiments that we have done for this project. We consider some possible causes for this increase in intestinal permeability, and which later I would like to discuss more. One of them is a starch overload, because when we reduce the protein, the amount of grain and obviously starch goes up. Soluble NSP, again, associated with grain, goes up, and the structural component and digestive dynamics of these diets are impacted, so they can have effect on intestinal permeability. This result is in line with an earlier studies that we did, again, as part of another project that I led with the support of AgriFuture. Back in 2019, we published a study that we showed compared to a diet that had higher concentration of protein and amino acids, the low protein diet had higher permeability, and this result has recently been observed with even mice-based diet. in US, and in this study by Leo et al. In 2023, they observed again an independent response to reduced protein diet and increasing the intestinal permeability. So we are seeing some consistent response into the reduction of protein for the gut permeability. So as I mentioned earlier, so there are some possible contributing factors. When we reduce the dietary protein, starch and grain goes up, and my excellent colleague, Dr. Sonia Liu, has done some work in that space with the starch, and they have seen it that once you cap the starch in a reduced protein diet, you can minimize the negative impact of those diets on performance. So we tend to think that starch is an area that needs to be further investigated within the context of reduced protein diet and also gut integrity, which I'm particularly interested. Another aspect that, again, is interrelated to starch is the rapid absorption of glucose and their competition with amino acids at the enterocytes, and also changes in the fiber and structural component of the diet that needs to be investigated. One of the things that we looked at were the expression of the genes related to clothing, the different clothing tight junctions and other tight junction proteins, and the genes related to intestinal integrity. I'm just putting up one of them here. Clothing 2 is a channel forming tight junction protein. Usually its expression, if it's increased, that's the indication of a leaky gut or compromised intestinal barrier. We can see here consistently in the birds that receive the lowest level of protein, they had higher expression of this particular gene compared with the birds that receive normal diet, and there was no difference between meat and bone meat containing diet and all vegetable diet. Another factors that is considered important in the context of gut health, of course, is the microbiota composition. And again, we observed that the richness of the bacteria was reduced in those birds that were either challenged or not challenged. So that was another indication of compromised gut health in those birds, because usually when you have a higher richness, it's regarded as a good sign to support optimal gut health. But that's not always the case, but usually when you see a reduction in richness, that's can be an alarming factor. The composition, overall composition, was impacted by reduced dietary protein, but not so much by the challenge. And when we look at the phyla bacteria, we observed that some group of bacteria, including proteobacteria, was changed. And in the birds that received lowest level of protein, excuse me, had a higher level of abundance of proteobacteria, which is regarded as a biomarker or a marker of gut dysbiosis. Another thing that we observe, which is noteworthy, is the overexpression of glucose transporter, which are shown to be having a link with intestinal permeability, and that is something that can be looked at further in terms of its potential to contribute to gut health in these kind of diets. move on from that study, we looked at another study that we were interested to look at glycine and serine and additional threonine in particular, follow on from a study that was done by Crystal et al. In 2020 at the University of Sydney, and they saw a positive effect of additional threonine in performance of birds fed corn-based diets. So we wanted to look at this in the context of gut health and the performance in these reduced protein diets, and see if we could see any positive impact. Of course, we used wheat-based diets. Although when we look at the performance, there was no particular benefit of additional threonine for the body weight gain, and for the FCR, only glycine and serine, as expected, was effective to reduce the FCR, which is a positive thing. And for other factors like intestinal permeability, we also did not see any differences between our treatments. Only we saw we saw a response on the challenge that we had. Another study, we looked at branched-chain amino acid based on their known impact on protein synthesis and being used as an energy substrate, their impact on reducing adiposity, which is another implication of reduced protein diet, intestinal immunity, microbiota, and so on. So we looked at two, again, diets: reduced protein diet, standard protein diet, and then we changed the level of branched-chain amino acids from -10% to 20% extra, and we kept the ratio between these amino acids similar to avoid possible antagonistic interaction between these amino acids. Again, when we look at the performance, we couldn't see any significant effect or improvement as a result of increasing branched-chain amino acids concentration in those diets. And based on what other researchers have found and what we have seen in this particular study, we think that even though we kept the ratio of these amino acids similar to the protein, to the standard protein diet, again, unavoidably, when we add the synthetic amino acid, the unbound amino acid ratio to the protein bound amino acid differs between these treatments, and that may still impose some opportunity for interaction between these amino acids to reduce their impact for improving the performance. What we found interesting in that study was that when in the birds that we had lower amount of branched-chain amino acids, we had higher permeability, again, independent of the challenge, but increasing amino acid, branched-chain amino acid had no effect on intestinal permeability. And just a couple of more slides. We looked at also microbial composition. There wasn't any significant effect, but something that was quite remarkable when we look at the composition of microbiota was that almost all these samples that we analyzed, they were highly dominated by firmicutes that suggested that there was some level of dysbiosis in the birds that received the reduced protein diets, again, somehow in line with what we had seen in the first study. So these are the factors that need to be, again, revisited and some tailored solution be developed for them to address these issues. Some take-home messages. I guess from this project, we have concluded that reduction of the erythropoietin has got profound impact on gut health of chickens, and that is largely independent of the challenge that we used in this study. And despite the common perception that if you reduce the dietary protein, you can expect the gut health to improve, the results of this study and that of recent literature suggest to the contrary. And it appears that once we actually meet the specification of amino acids matching standard diet, it is unlikely that manipulation of individual amino acids can improve gut health or performance per se. It may need to be in combination with other manipulation of the diets and strategies that can be tested in further research. And we think that further research really needs to focus on overall composition of these diets, particularly excess grain, starch, and fiber component, how they interact together, and what solutions can be come up with to improve performance and gut health of these birds. And thank you again for the opportunity. I'm happy to take any questions at the end of this webinar. And I'd like to thank the team, our team at SARDI that have helped me with this particular project. And with that, I conclude my talk. Thank you, Sarika. Thank you, everyone.
Thanks very much, Reza. We have received one question in the chat function, and I know I said I'd leave questions till the end, but because there was a lot of content and more content to come, I might go a little bit rogue, and I'm going to ask you this question now. So the question received, it was related to the part of the presentation which detailed the first study, and the question is if the crude protein reduction resulted in decreased feed intake.
What we have seen in that study, the birds actually adjusted their intakes in a fashion that actually did not result in a significant difference between the treatments. So it appears that they do compensate that reduction of dietary protein to some extent, because we didn't see any reduction in the feed intake, and their impact was basically was seen as part of the body weight and the feed conversion ratio.
Wonderful. Thanks very much for that. For everybody on the call, I will move to our next presenter, but if you've got any questions related to Reza's research that does come up, feel free to pop them into the Q&A chat function, and I'll get the team to share the paper that was snipped or screenshotted in one of Reza's slides, which we had the QR code to, which will provide a little bit more detail as well. And so with that, I'm going to move over to our second speaker, that is Associate Professor Sonia Liu. So Sonia is the Associate Professor in Poultry Nutrition at the School of Life and Environmental Sciences at Sydney University. Sonia is a nutritionist who designs practical poultry diets to meet global challenges in food security, sustainability, and environmental change. She's passionate about finding solutions for efficient and sustainable poultry production in different production systems, and explores this through her research interests, which include reduced protein diets, digestive dynamics of nutrients, enzyme application, feed grain quality, and broiler and layer nutrition. So during this next segment, we're going to hear from Sonia about the impact of climate on sorghum utilization in poultry diets. Over to you, Sonia. Thank you.
Thanks very much, Sarika. Good afternoon, everyone. Thanks very much for joining us and giving me the opportunity to share my research, our research with you. So this is a collaborative effort from colleagues across 3 universities and 2 RDCs. So the job I'm going to present to you is our recent research in the better sorghum varieties is under development at the moment. So I will get straight into it. So the idea is, so Australian sorghum has been under the impression it's going to cause inconsistent or even suboptimal growth performance for broiler chickens. And they triggered a series of research effort in the past 10 years at University of Sydney, but also at other universities as well. So one of the efforts derived from this early research is the protein in the dominant protein, caffeine in sorghum, is very hard to be digested by monogastric animals. Therefore, it triggered this breeding program to look into how we can improve protein utilization in sorghum for chickens. So there's two common challenges when it comes to design and feed ingredient evaluation studies. So the option one is when we fix the test grain inclusion rate, we are really testing the advantage, the variation of gross performance by changing the nutrient concentrations in the diet. And the second approach is we sort of keep the nutrient specification similar across all the diets. At the same time, we try to minimize the dietary composition variations, but there will still be some dietary composition differences. And in that way, we're hoping we test no damage principle, so it doesn't really compromise gross performance. At the same time, the diets give you feed cost advantage. So we sort of utilize both strategy, and both have its pros and cons. So we conducted two experiments. The first experiment was funded by GRDC through our collaborator, Professor Ian Godwin's group based at the University of Queensland. So this is their first attempt to evaluate several genetic lines of sorghum they were developing, and compare that against two commercial lines. So in this experiment, we had several treatments. So each sorghum variety is one single treatment, and their corresponding crude protein level as is basis that's been listed here in the table. Also, I have to mention, despite this as is basis, the dry matter content is quite consistent across board because this is grown in-house. These sorghums were grown in-house, so that was well controlled. And also the how they grow and during that process, that's well controlled as well. And so we used the offsex Ross 308 male birds, and we evaluated the performance and also digestibility as well from 0 to 21 days. And the reason for that is because we only have enough quantity to go to test that period of production, which is always a challenge when it comes to evaluating a new variety or trying to grow ingredients in a controlled environment. So this is what the diet composition looks like. And in this experiment, we try to fix the sorghum inclusion level. We let the nutrient specification concentration change during this process and to evaluate the differences in gross performance. In order to balance that big protein difference in the diet, called the big protein difference in sorghum, and we introduced another protein component, which is casein. As you can see, we kept the soybean meal consistent as well in this experiment design. This is no way saying it's perfect. We know casein is not being used commercially. However, we have to introduce another source of protein to balance the diet. So the results are sort of interesting. We didn't say sort of significant variations between different between all these diets. However, we did see correlations between, say, weight gain and also casein inclusion levels, despite the casein was included at a very, very low level. And this makes us realize for subsequent studies, we really need to adopt approach 2. It's both approaches again have pros and cons. So really it's really hard to determine which one to use. So for the first one, we tried the approach one. However, what we did find interesting though is we selected 3 sorghums that numerically had higher energy efficiency, higher protein digestibility, and also lower feed conversion ratio. And these 3 sorghums highlighted, we decided to go into next stage of evaluation. So that comes to investigation number 2. And this is a study funded by AgriFutures. We look at 8 different sorghum varieties and compare them against each other. So that blend there is just a blend of everything, and which I will show you why we did a blend. So again, so the aim of this second study is to further evaluate the impact of sorghum varieties, how that influence growth performance, and also nutrient utilization. For all these sorghum derived here, we analyzed the total amino acid profile for 16 amino acids. So that excludes tryptophan and cysteine. So what we found is we also can calculate, based on these values, we can calculate caffeine index, which based on the literature has been suggested to be a very good indication of the total caffeine concentrations in sorghum. So we want to verify this claim. So we our collaborators at Macquarie University, they actually quantified the actual caffeine. That's the total caffeine. So it's alpha, beta, and gamma caffeine all together. We couldn't really separate them down to alpha, beta, and gamma, but then with the overall caffeine information, it still shows quite promising correlation between caffeine index and also analyzed caffeine level. So what this means is, if we want to get a brief understanding of the level of caffeine in the sorghum, we can just test the total amino acid concentrations of sorghum and calculate the caffeine index and use that. That will give you a pretty good idea the actual caffeine concentration in the sorghum. Another interesting observation, just talking about sorghum per se, not the diet, we're not there yet, is the nice correlation between analyzed caffeine concentrations and also the crude protein level. This is consistent with what we see in the literature. So their previous investigations, not just in Australia, but overseas as well, showing sorghum contains a high crude protein, will contain a higher level of caffeine as well. That became a worry because caffeine potentially can be less digestible. However, in the study we conducted here, we didn't see compromise of protein digestibility, which is quite encouraging. So talking to our sorghum breeder collaborators, and they did do some magic at the breeding process to improve. In principle, that should improve protein digestibility. So this is just the overall comparison. If we just lump all these new sorghums, or we say better sorghums, compare with the commercial sorghums we had in this study, and this is the difference. So the high protein sorghum contained very high protein, and also the caffeine proportion increased as well, which kind of makes sense because caffeine is the dominant or storage protein for sorghum. The new sorghums contained about 10% more caffeine. However, the amino acids of caffeine itself actually did not change too much. That itself is a very interesting observation. So what we did is, what the Macquarie Uni, our collaborator there, they did is they quantified the caffeine, they separated the caffeine, and also measured the amino acid profile of the caffeine per se. So the reason why the amino acid profile of caffeine did not change is very good news, because there are 3 different kinds of caffeines: alpha, beta and gamma. And the beta and the gamma, they are the structural ones were wrapped outside the alpha caffeine, and the alpha caffeine is the storage protein, and the beta and gamma is the structural protein. So a lot of, say, early investigation showing the poor digestibility of protein in sorghum that comes from disulfide linkage between beta and gamma caffeines. So, if there is no change of amino acid profile in caffeine, that's an indication of there is no change of alpha, beta, and gamma, no significant change of that. In terms of percentage share between alpha, beta, and gamma. So we think that's a pretty good thing. However, there is a limitation though, because we did not really quantify system concentration in these sorghums, because that requires a separate sort of analysis, and that is higher in beta and gamma caffeine in comparison to alpha. And then move on to the amino feeding studies, just checking the time. Animal feeding studies. So this is the approach we did. So this time we didn't fix the grain inclusion level. So we formulate the diet with identical energy input and also crude protein level and the amino acid density as well. So as you can see, if we formulate a diet with the same nutrient specifications and the dietary composition changed a lot, especially when you look at the soybean meal inclusion levels here, it really changed a lot. That's corresponding to the individual sorghum's protein inclusion. So high protein sorghum actually get penalized because the soybean meal inclusion level is low. So that's why we developed a blend, and to sort of narrow down, well, we draw each kind of sorghum evenly to form a blend, and then we sort of use a half of the blend and a half of the tested sorghum diet combined together and to form the final test experimental diets. If all of these is very complicated to follow, please read our paper. And the paper has been published and you can read the details of dietary formulation. Then, all diets contained phytase, and also all diets contained acid insoluble ash as the dietary marker. It's a case study again, also mainly because these sorghums also grow under controlled environment. Therefore, we can only derive enough quantity for us to do cage-related study. It's also fit for our purpose because we want to do nutrient utilization and also digestibility assessment as well. So this time we targeted birds a bit older, from 14 to 35 days. And then we also measured the carcass yields and also apparent digestibility. So this is the results. We grouped the new sorghums and the commercial sorghums together, and what we found is there's no significant difference between feed conversion ratio. There's a slight increase of feed intake and weight gain for commercial sorghums, and a slight increase of fat pathways in the new sorghums. Move on to the energy utilization and also nitrogen retention. So energy utilization, we can say the new sorghums has had slightly better energy utilization, AME to G ratio, and also had a slightly better energy AMEN as well. Then digestibility in terms of starch, there is no significant difference. This is distal ileal digestibility. So there is no significant difference in terms of apparent starch digestibility at the end of small intestine. However, if we consider the feed intake, everything all together, so the disappearance rate is slightly higher in the new sorghums. And then for protein, there is no significant difference between these two. I guess this is somewhat a good news because the diet still contained variations in soybean meal inclusion, the way we design the experiment. So we were really worried that the new sorghum, high protein sorghum, would be penalized by the fact the diet itself contained less soybean meal. However, we didn't really see that when we look at the protein digestibility. So somewhat give us a little bit confidence of these high protein sorghums. Yes, it's high protein, it's high caffeine. However, the protein digestibility is still good. So this is my second last slide. So this is just showing 30% of dietary protein is derived from sorghum. And then when we look at the apparent ileal digestibility coefficients of amino acids so TG is the new sorghums, and the CH is the commercial hybrid. So what we see is quite comparable, or if it's not better. So it's actually pretty good. So here we just quote Professor Ravi's old work. So diets based on commercial hybrid sorghums, they advantaged because they had a higher level of soybean meal inclusion. However, when we look at the overall amino acid digestibility, and the new sorghums are quite comparable. So overall, the mortality rate in this study is very low, and it was not related to treatment, and there was no significant differences between dietary treatments if we just look at every single treatment, because the diets were formulated based to control the nutrient concentrations. So the benefit there is there's no drop for growth performance, and then it's up to the nutritionist in the field to say to do the cost calculation of the feed, whether utilizing so high protein sorghum at the same time reducing soybean meal inclusion would bring you benefit. So I would like to thank the collaborators, the founders, and also the colleagues at Camden, our technician team. So thanks very much for your attention.
Thank you very much, Sonia, and I'll get you to keep your video on, because there was a question that came through. There has a question that's come through that relates to your talk, and I'm trying to recall, I think it relates to that experiment when you were talking about or detailing experiment 2. And the question is, did you use wheat or corn as the control diet? And if not, did you expect, or were you expecting that chicken performance with sorghum would be similar to that with a sorghum or corn grain?
So in that experiment, we did not have a control in terms of using wheat or corn. However, we did include Liberty sorghum. So Liberty sorghum is a low protein sorghum containing about 8 to 9%, and it's a white sorghum. So from our previous work, the performance, if we formulate a diet based on Liberty sorghum, is quite comparable. That's because the diet would contain higher level of soybean meal, would have less phenolic compounds, and then so it will be comparable. And the overall the performance, they all outperform the ROS 308 performance objectives. Yeah.
Wonderful, thank you very much. And to the person who asked the question, I hope that did answer the question there. Otherwise, feel free to contact the team or Sonia directly if you know her personally to get more information. We will make sure that we share the resources. So that's the article or the journal paper mentioned in Reza's talk, as well as the one referenced in Sonia's talk with the post-webinar communications. And Reza, fantastic, because I've got a question for you, or a couple of questions related to your talk. And the first one might be a bit more broad and generalistic, but in one of the slides you'd mentioned that the richness of microbiota, it's an indication or a sign of optimal gut health. Just wanting to ask what else is generally perceived as optimum or gut health, or indications used to determine optimum gut health?
Yeah, thanks, Erika. Look, when we talk about gut health, we usually think about it as a 4-pillar main area, as I mentioned in my talk, is the intestinal barrier integrity, and we would like to see the integrity of the gut not compromised, and so we don't have a gut leakage and bacteria translocation. Another thing, as you mentioned, is the robustness and richness of the microbiota, and basically the abundance of beneficial bacteria that support digestion and immune functions and interact with the host organs. And another thing is the immune system. Of course, immune system is there for a reason to fight disease and pathogens and all those issues. But overactivation of immune system is something that we would like to see avoided, and basically we would like to reduce the overactivation, because that costs animal the energy and of course compromise the performance. Another thing is the oxidative status and hemostasis in the animal. So when we have the high level of reactive oxygen species, that's not perceived to be a good thing, because it damages the cell functions. So all these four pillars need to be in harmony, and I guess work in an optimal condition to for us to have an optimum gut health. We don't have a single measure for it, but we would like to see all these factors work nicely and coherently together to animal to have optimal gut health.
Thank you. Thank you very much for that. It's really helpful to have the parts of what you presented in the introductory part of your presentation sort of elaborated on. I've got another question for you, Sonia. There is a question for you in the chat, but I've got another one for Reza. So I might, once I've got you, Reza, off me, I'll ask you that one there, and then I'll jump to the other question we've received from the audience. So, Reza, for you, what other nutrients or ingredients could influence performance and gut health of broilers fed reduced protein diets, in addition to anything mentioned in your presentation?
Yeah, look, I personally think that there would be a lot of scope to optimize these diets by targeting things like component of the starch, not the starch in general per se, but the component of it and the rate of the starch digestion in comparison with the situation that we have a higher or lower digestion rate, and also testing some solutions and additives that can specifically target the intestinal health and microbiota composition, things like probiotics, but specifically the ones that can increase the microbiota diversity and help these birds to thrive better. And the interaction between these components, I think, is something that we have very little knowledge about it. And I think when we look at these reduced protein diets, similar to many other feeding regimes, there's not a single solution for that. It's a combination of factors that need to be investigated in future research.
Thank you very much. And that's exactly it. When you see sort of chicken feed, there is a lot of research that goes, there's a lot of research, a lot of complexity, and a lot of interactions that goes into sort of determine the best formulations. Sonia, a question for you that came through our chat was, how did you get the AME? So that's the apparent, for anybody on the call, apparent metabolizable energy values of high protein sorghum.
Yeah, so that's a very good question. Another challenge of testing a new ingredients, you just are not sure about the matrix value you're using. So in this experiment design, that's why we see the variations between the measured AME and also the actual formulated AME. So at the formulation stage, The AME was derived from LIR scan of the commercial hybrid. So these new sorghums, obviously, they're outliers. When it comes to the scan, that's for feed formulation. And then obviously subsequent nutrient utilization assessment, we did total excreta collection for 72 hours and to quantify the AME value.
Fabulous. Thank you very much, and thanks to those who'd putting questions into that Q&A or that chat function. Look, as we're approaching 5 minutes to the hour, I'll sort of put a hold on taking any new questions, but if you do have any other questions, feel free to email the program. We'll make sure in the post-webinar email that you guys get the email address for the Chicken Meat Program, or if you have my email address directly, you can send them through to me and I can liaise with our presenters here. So in our closing, I would like to bring your attention to the post-webinar survey that my support team will pop up onto the screen very shortly. I do mention it at the end of every webinar, but we do greatly appreciate your time in both being here, but providing that feedback, and it's so that we can make improvements to the upcoming webinars part of the series. In previous webinars, we do look at the responses received, and at times there has been suggestions to have more nutrition-based content, hence today's webinar. So we do like to listen to what has been provided in that feedback. A quick reminder, and I know we've said it a few times during the presentation, we will provide links to resources mentioned. And as always, look, please remember to report anything unusual or concerns about potential emergency animal diseases to the EAD hotline. It's very important that anything and everything is detected early. I would like to extend like a big lot of appreciation to our presenters, Reza and Sonia. We are really thankful for your time in presenting today, but working with us to prepare and present today's content, and the generosity and all of the knowledge that you've shared from the research that you've conducted. And of course, thank you to everybody that's shown interest in the webinar series, taken the time to be here with us today and as a part of other webinars. Our final webinar and I can't believe I'm saying this, the final webinar for 2024 will be an information session about the Gary Sansom Scholarship. And this is a great chance for me to share with you the applications for the 2025 scholarship opened just a few days ago. Again, link will be made available in the post-webinar comms, which provides further details on who's eligible. However, look, if there's anybody on this call, whether you're a researcher, whether you're somebody from industry, and if you supervise students who are eligible, or if you know somebody who would be interested and really passionate about the chicken meat industry, please spread the word, encourage them to join that next webinar where they've got that opportunity to hear more about the scholarship, but ask any questions about that application process. And look, with that, I'd like to wish you all a fantastic rest of your Thursday and rest of the week. And hopefully we will see you next time round. Bye for now.